Hello, my name is Pallas Burhen, and I'm a student in Jeffrey Gottlieb's lab at UW-Medicine's Center for Emerging and Reemerging Infectious Diseases. Firstly, I want to thank the organizers of the annual Undergraduate Research Symposium for giving me the opportunity to present my work. I'm going to be discussing an approach for evaluating antiretroviral drug resistance in human immunodeficiency virus, HIV-2, Group B. HIV is divided into two genetically distinct types, HIV-1 and HIV-2, that arose from cross-species transmission of simian immunodeficiency virus, SIV, to humans from chimpanzees and Sudi-Mangabe monkeys, respectively. The genetic divergence can be seen at right on the phylogenetic tree, with the two types circled in red, where the fewer branch points between termini on the figure, the more closely genetically related two species are. The genetic distance between types of HIV has certain implications for their biological properties, pathogenicity, transmission, and antiretroviral treatment. Within HIV-2, a total of 9 groups have been detected genetically, with only 2 groups, A and B, that have commonly caused infection in humans. Both HIV-2 groups A and B are endemic to West Africa. However, Group B remains the less studied of the two groups, with little known or published on drug resistance in the literature. Currently, there are no Group B clones in the National Institute of Health AIDS reagent program, and no other labs working on HIV-2 have an infectious Group B molecular clone available. The goals of my project are to construct a full-length HIV-2 Group B replication-competent molecular clone, 2, to build drug resistance mutations found in the literature for HIV-2 Group A into the full-length molecular clone of Group B. And third, to compare the resulting drug resistance data obtained for Group B to data published in the literature for Group A. Initially, my approach was to repair a defective EHO clone, shown on the right, which is entirely Group B. However, after several attempts at repair proved unsuccessful, my next approach is to use 7312A, shown on the left. 7312A encodes an HIV-2 Group AB recombinant virus. However, it is only Group A in the envelope gene in orange, which is not applicable to my project. I will build in common site-directed mutants thought to be important for drug resistance using site-directed mutagenesis. The general method uses a full-length infectious molecular clone of HIV-2 Group B that is used to transfect replication-competent cells. Virus is then titered and drug sensitivity assays are performed. The data from these assays is then analyzed and can be compared to data sets published for HIV-2 Group A. The next steps include continuing with site-directed mutagenesis with 7312A as a viral template, and characterizing drug resistance in Group B. I want to thank my project mentors, principal investigator Dr. Jeffrey Gottlieb and Dr. Robert Smith, senior research scientist in the Gottlieb Lab, for their guidance and support in my research project. I also want to thank Dana Rauji, along with other students in the lab. And finally, the Mary Gates Endowment for Students for funding my project, as well as the funding sources that support the Gottlieb Lab research. Thank you also to the audience for attending. I'll now take questions.